This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
And over to you, James.
Thanks, Aaron. So thanks everyone for coming to Kim's confirmation talk this afternoon. We were very fortunate to have Kim do her honours year with us last year, looking at some of the questions that are here. And as with all good research, she answered a lot of good questions and was able to ask a lot more good questions, which we were then fortunate enough to be able to keep her on to do a PhD. So Kim has a science degree with honours and an arts degree earlier than that, and it found us interesting enough to stay. So that was something we're very thankful for. So without any further ado, the other person I would like to thank is Ina Smith, who's online from Canberra, and I'm sure Kim will mention Ina more in the next coming minutes as well. So welcome, thank you for coming, and I'll hand over to Kim to talk about her PhD proposal.
Okay, thank you. And thanks everyone for coming to my confirmation. So I'll be going over my proposed project, which is looking at how foals respond to the Hendra virus vaccination. And just to start with, I'll give some background into Hendra virus, which causes a fatal disease of horses and humans. And I'll go through some key events in the Hendra virus timeline. So Hendra virus was first isolated in 1994 after an outbreak of an unknown illness among horses at a stable in Hendra, which is a suburb of Brisbane in Queensland. So the horses, they suffered from a severe respiratory illness, which was highly fatal. And during this outbreak, it also affected 2 humans who had close contact with the horses. And this resulted in 1 death. So investigation of the outbreak led to the isolation of a novel paramyxovirus. And it was initially classified into the morbillivirus genus, although it was noted that the relationship to other members of this genus was distant. And then an incident of horse and human illness, which occurred about a month earlier than in Mackay, was also retrospectively attributed to Hendra virus. And in this case, 2 horses had died, and a horse trainer who had assisted in the postmortems was hospitalised with meningitis after having contact with the horses. And this person had initially recovered, but then died the following year after a relapse of encephalitis. And preserved samples from the horses in Mackay, as well as the trainer, showed evidence of Hendra infection. So although these 2 incidents occurred within a couple of months of each other, they occurred about 1,000 kilometres apart. And so the source of Hendra was considered spillover from a wildlife reservoir, possibly bats, who could migrate and travel over long distances. Sorry, something's popped up. Okay, so from 1998 to 1999, there was a highly fatal outbreak of encephalitis among pig farmers in Malaysia. And isolates taken from these patients reacted strongly to Hendra antibodies. So this outbreak was also shown to be caused by another novel paramyxovirus that was closely related to Hendra. So this new virus was called Nipah. And the illness in humans was linked to an outbreak of respiratory illness in pigs, which was also attributed to Nipah virus infection. And then since this initial outbreak, Nipah has not occurred again in Malaysia, but has occurred several times in India and in Bangladesh. So the close relationship between Hendra and Nipah led to the creation of a new genus called Henipavirus, named after these two viruses. So in 2000, a paper was published which describes the isolation of Hendra from Australian flying foxes or fruit bats. So fruit bats had been suspected as the wildlife reservoir of the virus, and antibodies had been detected in all 4 of the fruit bat species in Australia. And so it was the isolation of live virus from fruit bats that supported them as the reservoir species. And then throughout the early 2000s, there were more sporadic outbreaks of Hendra in Queensland and northern New South Wales. Significantly, in addition to the human infections and fatalities in the mid-90s, there were further human infections in addition to the horse infections during this period. And just as shown by the annotations I've put in, Hendra is particularly dangerous for people whose work involves caring for sick horses, so particularly vets and stable workers. And then in 2011, there was a large spike in the number of Hendra infections, and this involved 18 separate spillover instances, which affected 24 horses. In 2012, an equine subunit vaccine became available on a restricted permit, and then it became fully registered by the APVMA in 2015. So while cases of Hendra infection in horses has continued to occur pretty much every year since then, there has not been an infection in a vaccinated horse. So all infections have occurred in unvaccinated horses. And one particular case of Hendra that is of note is an infection that occurred in a horse in the Hunter Valley region of New South Wales. And the reason I bring up this particular case is that the Hunter Valley is an important region for the horse industry. So there's quite a lot of horse breeding farms, as well as other equine related activities in that area. And recently a new variant of Hendra has been identified, and this is called HEV-G2. So actually a horse infection in 2015 and another in 2021 has been attributed to as this variant. And also the variant has been found in the grey-headed flying foxes, which are present, which are present around Melbourne and Victoria. So Hendra virus is a paramyxovirus. It's a large family of viruses that include many important animal viruses, such as rinderpest, canine distemper, Newcastle disease, but also includes other important human viruses, such as measles. So all paramyxoviruses have a non-segmented negative sense single-stranded RNA genome. And Henipaviruses have a longer genome length compared to other paramyxoviruses. So when Hendra was first isolated, it was described as having a double-fringed appearance. And this is due to two types of surface proteins. So the glycoprotein, it facilitates attachment to a host cell, and fusion protein for cell entry. So there is an additional 4 proteins, the matrix, phosphoprotein, nucleoprotein, and the polymerase. And Henipaviruses are also distinguished by a broad host range. So several more Henipaviruses have been identified since Hendra and Nipah. So these occur in several countries, and they have been found in several animal species as well. So this tree is from a paper describing Langya Henipavirus, which was in the news earlier in the year. So it was discovered in human patients in China and then also found in shrews, which are the possible reservoir. And there are also 2 Henipaviruses that have been found in Korea, also have been found in shrews. and they're called Gammak and Daeryong henipaviruses. And rats are suspected as the reservoir species for Mojiang henipavirus, and fruit bats are the reservoir species for Hendra nipa, a henipavirus in Ghana, as well as another Australian virus called Cedar virus, which is thankfully a non-pathogenic henipavirus, so can be a useful tool for henipavirus research. So horses become infected with Hendra from contact with the bat secretions, and then infected horses can then pass the virus on to other horses, as well as to humans via close contact with body fluids. So all 7 cases of Hendra have had close contact with an infected horse, and no direct bat to human transmission has occurred, and also human to human transmission has also not occurred. In contrast, for Nipah virus, direct bat to human as well as human to human transmission has occurred. But for Hendra, this means that horse vaccination not only prevents horses becoming infected, it indirectly protects the humans from infection as well. So the Hendra vaccine is a subunit vaccine. It contains a soluble form of the Hendra G surface protein, and it is effective in preventing disease as well as viral shedding, which is important to prevent ongoing transmission. So the current protocol for vaccination is an initial 2 vaccinations given about 3 to 6 weeks apart, followed by the 6-month booster, and then annual boosters thereafter. So foals are recommended to begin vaccination from 4 months old, or from 6 months old if their mare is vaccinated. And this is due to the maternal antibodies that are transferred from mare to foal that may have an effect on vaccine response. So several studies have been published regarding Hendra vaccination in adult horses, but no studies have been published regarding Hendra vaccination of foals. And vaccination response can be different for foals. So just a bit of an overview, there's no placental transfer of antibodies that occurs during pregnancy in horses. So foals are born with no antibodies. And it's the colostrum, which is the first milk that's produced by the mare, that is the only source of antibodies. And the colostrum must be consumed within about a day after birth, as the cells of the small intestine will start to mature and then can no longer absorb the antibodies. So hopefully, if the foal has had colostrum within the correct time, the antibodies absorbed will circulate and then gradually reduce. So it's thought by about 5 to 6 months of age, these passive maternal antibodies will have disappeared. Therefore, the recommendation for foals to begin Hendra vaccination from 6 months old. However, there's still many unknowns regarding foal immunology, and this is summarised well in a 2015 review paper. So particularly, foals overall respond poorly to vaccination. And although several studies have looked at maternal antibody interference as the cause of this, it's still not really definitive. In addition, there is incomplete understanding of other immune components that are transferred from mare to foal, and just the overall knowledge of cellular immunity in the foal is incomplete. So I'll just go over the study aims of the project. And I do have 3 major study aims for my PhD project. The first is the detection of Hendra-specific antibodies in horse serum using various methods, including a Luminex microsphere assay and comparing with methods that measure neutralizing antibodies. The second aim is to characterise the foal response to Hendra vaccination in foals both from vaccinated mares as well as foals from unvaccinated mares. And the third aim is then to investigate the factors that may affect the response to Hendra vaccination. So particularly the effect of maternal antibody interference, and also might look at the cellular immunity components. If there is a possibility of asymptomatic Hendra infection that could be affecting the antibody response, and also possible effects from other routine fall vaccinations that are also given. So now I'll go over the methods I'll be using to measure the antibody levels and the neutralizing antibodies for AIM-1. So there are 3 methods that I plan to use in this part of the project. So each have like certain advantages as well as disadvantages. So the first method is virus neutralization testing, or also known as serum neutralization testing. So VNTs were used during the vaccine efficacy trials, and they are useful because they give a neutralizing antibody titer, so they can show protective levels. So the vaccine studies showed that titers as low as 32, possibly as low as 16, were shown to be protective against Hendra. However, the VNTs require a 3-day incubation time, so it is time consuming to perform. And it also requires the use of infectious virus, which is difficult for Hendra, as it means that the VNT must be performed in BSL-4 conditions. So this makes it an expensive test to have done. There are safety issues with using the infectious virus, and the time-consuming nature of the VNT makes it not as practical for large numbers of samples. The Luminex microsphere assay, it is a similar idea to an ELISA assay. So in an ELISA assay, the wells of a plate are coated with the antigen, but in the microsphere assay, it uses coloured beads that are coupled with an antigen. So in this case, it's the soluble G protein that's also found in the vaccine. So the antigen will then bind to the antibodies that are present into the sample. And then the detection antibody and a substrate is added so that the plate reader can read the colour signal. So the Luminex analyzer will detect not only the reporter molecule, but it also detects the signal of the beads. So it has multiplex potential. So when using different coloured beads that are coupled to different antigens. So the results of the Luminex are given as a median fluorescent intensity, or MFI. So the HEV Luminex assay is very time efficient. It only takes about 3 hours and can be performed in a PC2 lab, as no infectious virus is used. It's only the soluble G protein. And it's also beneficial because it only requires about 1 microliter of serum. However, the results don't give an indication of protection. So the MFI is the level of antibodies, but not of neutralizing antibodies. So earlier I mentioned the CEDA virus, which is a non-pathogenic henipavirus. And so the third technique involves a recombinant CEDA virus, which can be utilized for a surrogate neutralization assay. So in this study from America, a firefly luciferase gene has been inserted into the recombinant CEDA virus genome. And they use this to screen a large molecule library in order to identify inhibitory compounds that could be developed as potential henipavirus antivirals. So the plan is, so this assay can be used for indicating neutralizing antibodies. And unlike the VNTs, it can be performed in a PC2 lab. So it's like more practical and safer than the VNT. So the plan is to make use of all 3 of these methods. So testing all serum samples in the Luminex, then taking a subset of the samples to the US for the CEDA virus assay, and then also sending a small number of samples to CSIRO in Geelong for the VNT testing. And then so hopefully by using the result of all of these, we can determine protective MFI levels. So I'll go on to aim 2 now, which is characterising the foal response to vaccination. So this project will make use of a large number of horse serum samples that have been collected from a breeding farm in the Hunter Valley, which, as noted earlier, is an important horse region, so the horse capital of Australia, and where Hendra has occurred previously. So this particular farm began vaccinating their horses in mid-2019. So the first vaccination was given to most mares in June, followed by the second vaccination in July. And then horses give birth from August, so sampling started during this foaling season, and paired samples were collected from mares and foals at this time. So for the mares, this was 1 to 3 months after their second vaccine, depending on what month they gave birth. And for foals, they were sampled after they'd had colostrum. So mares and foals were sampled again approximately 3 months later during the foal branding period. So the mares were usually due for their third vaccination at this point. Then the foals were sampled at each vaccination point. So at the first vaccination, they were approximately 6 months old. At the second vaccination, 1 month later. And then again at their third vaccination when they were about 12, 13 months old. So mares and foals from the 2020 foaling group were also sampled at similar times. So for the mares, they were sampled approximately 7 to 9 months after their third vaccine, again, depending on what month they gave birth. And then during the branding time, they were usually due for their annual booster vaccination. And then foals, and then the foals were, so the foals were just followed at the same, sampled at the same points as the 2019 foals throughout their first 3 vaccinations. And I'll just note that the sample was taken immediately prior to the administration of the vaccine at the vaccine sampling points, except for the mare V4, where the blood sample was taken within a month either side of the vaccine. And just this table at the bottom is a summary of the sampling points, and I'll just refer to that later in the results. So a total of 3,661 serum samples have been received so far from this farm. And this is from 986 horses, including 410 mares and 256 foals born in 2019 and 291 foals born in 2020. So the Hendra vaccinations are recorded on a registry on the Zoetis website. So I checked all of the horses' vaccination history. So the majority of the mares were vaccinated for the first time mid-2019, with several of the mares vaccinated before this, even as far back as 2013. And then again, most of the foals born in 2019 were vaccinated throughout 2020, and the foals born in 2020 vaccinated throughout 2021. So a number of these samples have been tested already with the Luminex assay, and this has taken place at the CSIRO lab in Canberra, including a batch of samples that Ina tested during the sort of last of the lockdown period last year, which saved my honours project. So the samples of 121 of the 2019 mare foal pairs and 68 of the 2020 pairs had been tested, as well as some single samples from 41 of the dry mares and 21 samples from the foals born in 2018, who just still happened to be around the property when the sampling period began. So I'll start by looking at the transfer of maternal Hendra-specific antibodies. So this graph shows the paired mare and foal samples from the 2019 and 2020 foaling. So we can see the transfer of maternal antibodies from mare to foal, which is very efficient. So it also suggests that the Hendra antibodies are concentrated in the colostrum, which is consistent with like previous studies, although we didn't have any colostrum samples available to test. Because I have not completed one of the projects, which is intended to sort of compare the neutralizing antibody titers with the MFI results from the Luminex. For the time being, we're using the tentative MFI ranges that's used by CSIRO. So a 1000 MFI is the current conservative threshold. So an MFI below 1000 is considered negative. So 1000 to 5000 is considered a low positive, 5000 to 10,000 a medium positive, and over 10,000 is a high positive. So there was one mare-foal pair from 2020 that did have a negative MFI result. Otherwise, both mares and foals were above the 1000 threshold at the foaling time. So now looking at the foals only, again, there is the results from the after colostrum sampling with the large MFI values. And then we see a reduction of the maternal antibodies sort of 3 months later during the branding period, and a further reduction 3 months later just prior to the administration of the first vaccine. So 57% of the 2019 foals are considered negative by 6 months, and then 74% of the 2020 foals. And then minimal response to the vaccination is shown by the samples collected about 4 weeks after the first vaccine, with 38% of the 2019 foals and 72% of the 2020 foals are still considered negative. And then again, 6 months later, just prior to the administration of the third vaccine, we have 40% of the 2019 foals and 59% of the 2020 foals are still negative. So unfortunately, we do not have any post B3 samples so that we can look at the response to the 6-month booster vaccination. And now just looking at the mares. So the mares show a good response to vaccination. So remembering that the 2019 foaling sample was either like within 1 to 3 months after the mares had their first 2 vaccinations. So we can see the antibody response here, and the antibodies do drop by the time the mares are due for their third vaccination, although only 4% have dropped below the 1000 MFI threshold. And then the response to the third vaccination is good, as shown by the 2020 foaling sample, which was about 7 to 9 months after the third vaccine. And then at the V4 sampling point or annual booster point, it is higher overall, just because some of the mares being sampled before vaccination and some after. And then the looking at the MFI values now of the brood mares and 2019 and 2020 foals that we've already discussed can be seen in this graph alongside the results from the 41 dry mares and the 2021 foals born in 2018 who were still at the farm during the sampling period of the study. So both the brood mares and dry mares have higher MFI values as compared to the foal groups. Just looking at the small group of the 2018 foals, which show a sort of moderate response. So these foals were born before their mares were first vaccinated for Hendra, which was around mid-2019. So they did not receive any passive maternal antibodies. And they also began vaccination around mid-2019, so we're around 7 to 9 months old at the time of their first vaccination. So just based on looking at where the results of the 2018 foals are sitting on the graph, the response of B3 is not quite as good as the mares, but not quite as bad as the other foal groups. But definitely more analysis and possibly a larger sample size of foals with no maternal antibodies would be required to sort of reach a more definitive conclusion about the role of maternally derived antibodies. So considering the overall poor foal response to vaccination, I guess the question is it because of the presence of interfering maternal antibodies? Is it related to foal age and immune development, or is it a bit of both? And also, does a reduced response matter as long as the protective antibody threshold is met, which is why it is important to address the first aim of my project. So I'll now look briefly at the aim 3, which is to investigate factors that interfere with foal response. So the initial focus will be on maternal antibody interference. So to do this, we will vaccinate and sample mare and foal pairs where the mares have not been vaccinated before foaling. So we have recruited a farm in Northern Victoria for this study with about 80 mare foal pairs. And the plan is to begin vaccination and sampling of both the mares and the foals in February 2023. And ideally at monthly sampling points to get some samples between the vaccinations and as well as after the third vaccination to see the response to the 6-month booster. So the future plans for the study is to continue the Luminex testing of the horse serum samples, and then to take a subset of the serum samples to Maryland in the USA for the Cedar virus assay, and then also sending the smaller number of samples for BNT testing at ACDP at CSIRO. And then for aim 2, its focus will be on the vaccination and sampling of the mares and foals on the Victoria farm. So that will be throughout 2023. And ethics have been submitted for that. Aim 3, so initially it will be to focus on comparing the results from the New South Wales mare foal pairs and the Victoria mare foal pairs to investigate the role of maternally derived antibodies. And other options include using commercially available Luminex kits to investigate other immune components, testing the horse serum samples we have, as well as sourcing other horse serum samples to screen for Hendra antibodies and investigate possible rates of exposure, and possibly developing a multiplex assay to measure antibodies elicited by other vaccines. And I'll just take a minute to thank firstly my supervisory committee, Peter for stepping in as chair, James, Carol, Kirsten and Ina. Particular thanks to Ina and her colleagues at CSIRO in Canberra. They hosted me for a week earlier this year. Thank you to Christopher Broder, who supplied the Hendra G protein that's used in the assays. And my PhD scholarship is funded by the Australian Government Research Training Program. And thank you everyone here who attended my confirmation today. Here are my references, and thank you.
Thanks, Kim. That's excellent. Okay, does anyone have any questions for Kim? Don't all jump in at once. That makes it really difficult.
As I said, Kim, you have dazzled them
Oh, thank you.
You have dazzled them and you've been very clear and articulate, even for a Luddite like myself.
I think one thing that Kim didn't mention was that this morning she would have been notified that she's received a Barringer scholarship to help her get to Maryland. So that makes things a little bit even easier. So that's all good.
Congratulations. I'm sure everyone is so pleased for you. That's fantastic.
Yeah, I just got that email this morning. It was like, oh, I didn't apply for a scholarship, but
These things work in mysterious ways, Kim.
yes.
It's the miracle of Christmas. You've had a Christmas miracle, Kim.
Cool. No questions at all whatsoever.
They're all listening.
I
We had nearly 35 people today, and they're all there. But I think it was a very comprehensive presentation. I think, wasn't it, James?
I agree. So I think, Kim, we might let you off the hook and just say thank you, and thank you, Aaron, for putting in the acknowledgement of country, which I erroneously and egregiously left off earlier on. So thank you.
That
Kim.
was equally my fault, but we popped that in there. And as always, we do take that seriously, and we thank everyone and for forgiving our admission. And congratulations to Kim. I think we speak on behalf of everyone, and I'm sure your panel will be looking. Your committee will be looking very, very much forward to your research. And so on that note, I might end the recording there.